So if you did the enzyme experiments, then you probably did this one as well, which is how to immobilize an enzyme. Immobilized enzymes are basically any enzyme that is not free in solution. So they're not dissolved, right? It's not there in solution. What it is it's been trapped in a gel or attached to some sort of surface of a solid or a semi-solid material, right? So either it's inside or attached to some sort of solid or semi-solid material. Okay. Often that's a gel. If you've done this, then you would have made a gel beads using sodium alginate. So sodium alginate would have been the material that you use to immobilize the enzyme that you work with. Probably you immobilize cells rather than an enzyme, and the cells that you immobilize probably E cells. But the reason we immobilize the E cells is because we wanted to exploit their enzymes. And the enzyme that really we wanted to exploit was sucrase or invertase. So we'll come to that in a minute. So what's the big deal? Why would you want to immobilize enzymes? Well, because there's all kinds of advantages. So I'll just show you how this works. And they're used in what's called bioprocessing. Supposing you had your enzyme, and we'll talk about the enzyme sucrase. Supposing you just had the enzyme and or cells, right? But just the enzyme itself. Let's focus on that. So you take your enzyme, you mix it with some sodium alginate, you make your beads with sodium alginate, put them into calcium chloride, the beads harden, and you get this trapped enzyme, immobilized enzyme in the beads, right? Stuck there. And it doesn't matter whether you put those beads into liquid, they won't leave the beads. They're attached to and inside the bead. Okay. So if you fill a column, like literally it's a large, kind of various sizes. You can have lab sized ones, you can have industrial sized one, as big as a house. But they're basically like a big tube filled with these little beads. And if you pour into the top, you pour a sucrose solution, then as the sucrose trickles down through the beads, the enzyme will catalyze the change of that substrate, which is sucrose, white table sugar, into table sugar. Sucrose, if you don't know, it's a disaccharide, right? And it's composed of a glucose and a fructose. So what sucrase does is it breaks that bond between the two monosaccharides. So you get the two monosaccharides coming through out of the bottom as product. And what they call that is glucose fructose syrup. You've probably seen it if you look on the labels of lots of processed foods, and you'll see glucose fructose syrup. Sometimes you see it called high fructose corn syrup. It's the same kind. It's all made by this kind of process, right? So bioprocessing is used for lots of industrial purposes, but one of them is for making food additives, right? And this is one, glucose fructose syrup. Why would you bother? Why would you not just add the sugar? Because it turns out that sucrose isn't actually as sweet as fructose. So for the amount of sucrose you put in, for every gram of sucrose, the gram of glucose fructose syrup that you get out is sweeter. So you need to use less of it to sweeten foods. So it's cheaper to use, right? Even though you've had to go through this process, which obviously costs money, but in the end, it's cheaper. So bioprocessing then is, broadly speaking, it's the use of cells or enzymes to make something useful or to break something down that is harmful, like waste. So you could use it for, like we just had there, glucose fructose syrup manufacture, using sucrase, but you could also use it if cellulose. There's no shortage of cellulose in the world. So there's some enzymes from fungi in particular that are able to break down cellulose. Like we can't do it. Most mammals can't do it. Some microbes can do it. Some bacteria, some fungi. So they've found that you can use these enzymes, cellulases, to break down cellulose, which is a polysaccharide. It's a chain of lots of glucoses. You can make sugar from that. Good trick. So what's the big deal? Why would you do it? So if you look at the list here, it's more economical because it can be reused. Just to go back to our picture. So you can keep pouring sugar on top and getting product out the bottom, and the alginate just stays in there in the bead. Sorry, the enzyme stays in the alginate bead. So you can just, it's continuous. You just keep continually using it. They're very stable, the enzymes, when you have them trapped in the bead like that. They don't tend to break down as easily, so they last longer, which is again makes it more cost efficient, right? So they're more stable. They can be used over and over again. But the biggest thing of all, to be honest with you, is because the enzyme stays in the bead, right? The product that comes out at the bottom is free of enzyme. So if you don't use immobilized enzyme, if you just added enzyme into your sugar solution, right? Suppose you had your sugar solution here and you wanted to change it into glucose fructose syrup. So you pour in your enzyme. Now the problem is, grand, you've got your glucose fructose syrup, but it's enzyme in there as well. Somehow you've got to get the enzyme back out again. And that's not easy. And it's not cheap. So the separation thing at the end to get rid of your enzyme that's contaminating your food thing, because you can't have enzymes in food, like I said before, because of possible allergic reactions, right? So that's a really important thing. It's much easier, in fact, it's not necessary to separate then the enzyme from the product. So that's really good. You just need to take out the beads, and sometimes you need to take them out because you just pass the substrate through the beads. Okay, so a bioreactor, that's the kind of vessels that you would do bioprocessing in, as well as growing bacteria, for example, in to produce something. It could just be cells, it could just be enzymes, right? Or some biological component. That's what we use bioreactors for. They're large stainless steel vessels usually. They are very tightly regulated and controlled, monitored and controlled. So they're looking, everything is just as it should be. And it's usually a computer somewhere that's running the whole show. There's all these sensors in it. Show you a picture. There's a little tiny lab one. It's a 1 liter one, right? But you can see all these, there's all these things sticking into it that are all linked to a control unit. Controlling what? Controlling pH, temperature, concentration of enzyme, concentration of substrate, the amount of waste that's in there, all that, whatever kind of stuff that you need to be considering. A bioreactor is the way to go.